Good morning, everybody. We'll be starting in 1 second. I hope everybody is doing okay today. My name is Piotr Slis. I'm a Chief Research Information Officer at Boston Children's and coordinating all the research computing activities. Shira is not here today. She had a conflict with a BCH COVID committee. It will be me as well as Ann Paduri, Director of the BCH Epilepsy Program, who will moderate the session and introduce the speaker today. We have some increased Zoom security today to not allow too many strangers into our channel. We are limiting the chat window so you can chat directly to me, to the host, and I can send the messages to the speaker. Also, if you have questions, you can raise your hand virtually, one of the icons here, and we can funnel you directly to ask the questions at the end of the session if we wanted to make it a little bit more interactive. Just a couple of general reminders. If you guys any computing research computing guidance, please visit our RC Wiki page. Just in the browser, type RC Wiki, and when you're on the BCH intranet, they will provide you with the overview of all the services which we provide. Or feel free to email research.computing@childrens.harvard.edu, and we can provide guidance as well. In particular, nowadays, we are expanding our service to provide general consulting. If you guys need to set up better from home, any other services, just email us. The series of Tuesday seminars started a couple of weeks ago. We have a very exciting lineup after today as well. April 28th at noon, we have a speaker from Karolinska Institute who will talk about epigenome-wide meta-analyses of blood DNA methylation. So that's next week. And then we have speakers from Wellcome Sanger on May 5th and May 12th from Harvard Medical School. And also starting in May, on Fridays at 2 p.m., we will be doing 4 meetings, our Genomic Innovation Symposium, which has been rescheduled from the in-person meeting. That will take place on Fridays at 2 p.m. More information about this soon. And we're looking always for your further input and suggestions how to set it up. So a couple of technical notes, as I mentioned before. If you have questions, raise your hand virtually or message me directly. And the seminar today is being recorded. So at this point, I want to hand it over to Ann Paduri, who will introduce our guest today and moderate the remaining part of the session. Thank you, Ann.
Great, thanks, Piotr, and welcome, Bekim. Everyone, thanks for coming, and it's really a pleasure to introduce Dr. Bekim Sadikovic from Western University. He is, as you can see, the division head of the Molecular Diagnostic Division of the London Health Services Center and Associate Professor of Path and Lab Medicine at Western University. Bekim has a rich background and numerous publications and research projects focused on genetic variants and also epigenetic markers of disease. Our work has intersected through the ClinGen epilepsy panel, where we're looking at curation of genetic variants related to epilepsy, but the world of epigenetics is still a bit of a black box for a lot of our disorders where we've just begun to figure out the genetics. And so now we have colleagues like Bekim who are taking us to the next level, getting into this really fascinating world of epigenetics. Bekim has looked at a number of different diseases, both inherited and sporadic diseases, cancers, neurodevelopmental disorders, and brings to us really a wealth of knowledge and experience, not only on the research side, but also from the practical side of actually interacting in a clinical lab setting. So really looking forward to hearing your talk on DNA methylation signatures and Mendelian developmental disorders.
That very kind introduction, Paduri, and thanks everyone for joining the call today. I'm excited to be able to share some of the work we've done in the last few years that's focused on epigenetics of hereditary disorders. I hope everybody's managing through these fairly challenging times, and I'm very happy to at least participate in these type of events. I've had the opportunity to do so in the last few weeks. It's a good way for the community to kind of get together and feel like we're socializing without actually being in the same place. So the talk focuses on specifically Mendelian developmental disorders and DNA methylation signatures in these disorders. The talk is kind of put together to really be fairly, I would say, a broad overview of some of the work we've done in the last 5 years. There's going to be a bit of, I'd say, kind of in-depth computational discussion and bioinformatics, but it's really a very generic talk. So for any of the audience, it's really interested in kind of diving deeper in some of the computation behind a lot of the work here. There's going to be references shared in here, so you can kind of get into that a bit more deeper. In my role, I oversee a diagnostic lab, and so I've always been, perplexed and challenged with genetic variation that we get when we sequence genes. One of the banes of the existence in a clinical setting is variants of unknown clinical significance. And this is the kind of mindset that I had, when we started really exploring this idea of DNA methylation and DNA methylation signatures. So it's really very, very practical and, say, clinically minded. And a lot of the work today is really focused on biomarker discovery and how we can use epigenomics to really help us in an immediate kind of clinical setting. There's a lot of interesting biology that seems to be coming out of this. That's certainly, I think we're, seeing the tip of that iceberg really right now, and many, many more years work to come to start kind of deciphering that. So what is epigenetics? Well, classic kind of definition of epigenetics is that it's a study of heritable changes in gene expression or cellular phenotypes caused by mechanisms other than changes in underlying DNA sequence. And I must admit that this definition doesn't really sit very well with me, because it's either a correct definition and all the rest of what I'm going to talk about today is not epigenetics, or it is that it's probably requiring some augmentation as a kind of a definition. But you'll see what I mean about it when I talk about it later on. Essentially, what it means is functionally relevant modifications to genome that do not involve a change in the nucleotide sequence. But one of the interesting distinctions about epigenetics versus genetics per se is that it can potentially be environmentally responsive, so micro or macro environmental responsive, and it potentially can be reversible or changed over time or as a response to environment and so on. It's not static, so epigenomic profiles in humans, for example, in our DNA rapidly change throughout lifetime. So primordial germ cells undergo a wave of demethylation, for example, followed by remethylation prior to fertilization, and again post-fertilization. And this has to be very carefully managed, these mechanisms, and any errors can result in developmental problems. Some of what we know about epigenetics is really focusing on errors of imprinting, which are essentially regulated at time. And essentially, really all we knew about epigenetics in constitutional setting was from the work that was done 20, 30, 40 years ago, specifically in imprinting disorders. And that's kind of where we stopped. And a lot of the work that's kind of been done since then in the world of epigenetics, DNA methylation in particular, has been done in cancer. And there's a plethora and huge body of knowledge of changes in biomarkers in epigenetics in cancer. But I think there's been a kind of a reinvigorated interest in looking at epigenomes of related to kind of hereditary or constitutional disorders in the past decade or so. The other important thing to mention is that unlike genetic sequence, different cells and different tissues in a body have vastly different DNA methylation profiles. And part of the reason why that is because DNA methylation plays an important role, and epigenetics in general, in regulating and organizing chromatin, and thereby eliciting or participating in the function of the chromatin, which is regulation of gene transcription to an extent, and the reflection of the differences in the phenotype in these different tissues is oftentimes mirrored in the changes in DNA methylation profiles in these different tissues. From a practical standpoint, and me wearing my now clinical hat on my head, from a laboratory diagnostic standpoint, unlike in a genetic setting where we can test for blood DNA or a muscle biopsy or an FFPE tissue from a patient, and rely on the fact that DNA sequence is the same across these tissues, that's not the case for DNA methylation profiles. So we're thinking really about moving this technology into a clinical diagnostic kind of setting. We really need to start developing databases that are going to be tissue specific. They're going to enable us to really make sense out of this data in a clinical setting. That's a big challenge. So I should say really that most of the work that I present for the rest of the talk today is really focused on peripheral blood, whole peripheral blood, and the reason why we chose that as a tissue is because it's the most common tissue that we get in a diagnostic setting. If we're looking to set up diagnostic biomarkers, we want to make sure we have the most accessible tissue. On a molecular level, essentially DNA methylation is an addition of a methyl group to a fifth possession of cytosine by a methyltransferase enzyme. DNA methylation profiles work in concert with other epigenetic mechanisms, including histone modifications. And ultimately, I guess maybe in very simplistic terms, epigenetics plays a role in chromatin regulation and compaction. So open chromatin or euchromatin tends to be mostly associated with transcriptionally accessible regions. So genes can be turned on in these regions, and these regions tend to be oftentimes hypomethylated, or alternatively, hypermethylated DNA tends to be more heterochromatic or more compacted and a bit less transcriptionally active. Now, these are generalizations, they're exceptions to these rules, but a simple way to think of DNA methylation or loss of methylation: gene activity is on, hypermethylation, gene activity is off. The outline of the talk today. I will discuss, through the talk and highlight a few of the key kind of studies that we've done over the last few years that exemplify some key points that are important to understand about this phenomena of these epi signatures and how they may apply in a practical setting. So first part of the talk, we'll talk a little bit about our work with imprinting disorders and Fragile X, and then we'll talk a little bit about some of the early work in neurodevelopmental syndromes and some of the complications we've seen as we expanded from single genes to gene complexes, and then really talk about kind of larger scale kind of cohort type studies, and some of the work we're now focused on is essentially we're starting a national clinical trial that's going to be testing thousands of patients using DNA methylation profiling to look at the impact of introducing this technology into a more kind of routine clinical setting. And I'll talk a little bit about some of the clinical testing that's already being done in US and in Europe using these technologies. So for the vast majority of the work I'm going to present today, we use Illumina methylation arrays. They started out as 27,000 probe arrays to then moved on to and expanded to 450K arrays, and then more recently grew to about 850,000 methylation sites, probes, covering vast majority of REF-seq genes and CPG islands in the human genome. And CPG islands are high-density CPG regions within promoters, about half of the genes in the human genome. Most of these tend to be housekeeping genes. Most of them tend to be hypomethylated. Essentially, it's an array-based platform that is very robust, very reproducible, and scalable. And that's why it's part of the reason why we consider it to be a platform of choice if we're thinking about scaling this in a kind of a production setting, if you will, in a clinical setting where we're required to process patient samples, turn around, and report results in a short period of time with not a very large cost. Certainly, there are other approaches genome-wide. By sulfide sequencing is probably the most comprehensive way of doing this type of work. I do think that at some point down the line, this type of work will evolve to be using those type of platforms. But currently, in its form, it's primarily array-based. So early work, simply, this is, when we started doing this work, I asked a simple question, that is, can we take a whole genome methylation array and replace a bunch of targeted imprinting assays that we're currently running in diagnostic labs? So imprinting disorders like Prader-Willi and Angelman in the diagnostic labs are commonly done and oftentimes require targeted assays that are either PCR or a restriction digestion based on the underlying context of DNA methylation profile and so on. We have number of these disorders, and this data basically just show you very simply visually a region in a SNRPN gene promoter that's imprinted, and in normal controls, purple lines essentially are all the normal individuals. They're hemimethylated. So imprinted regions are methylated in one of two alleles. So what happens essentially in a normal individual, you have 50% methylation. These are all these different purple lines of different normal individuals. Patients with Angelman syndrome have loss of the methylated allele, sometimes as a result of deletion, sometimes as a result of uniparental disomy, sometimes as a result of an upstream promoter or mutation in the imprinting controlled region as well. So we end up there is essentially hypomethylation, and the opposite is true for Prader-Willi. We end up with hypermethylation. So you don't need very sophisticated kind of bioinformatics to say that it's fairly easy to pick up patients with Prader-Willi and Angelman using a microarray-based approach. Another region on chromosome 11p is a region associated with Beckwith-Wiedemann syndrome. Unlike Prader-Willi and Angelman, Beckwith-Wiedemann often comes in a form of mosaicism, so certain percentage of cells do have it and other ones don't. And what you're seeing here in red lines are various patients with different levels of mosaicism for Beckwith-Wiedemann. We can detect down to about 10% mosaicism using this technique. There's an example of a patient with Silver-Russell syndrome, where the cause of this disorder is uniparental disomy. So essentially, the entire chromosome is inherited from one of the two parents, so two copies of it, and that ultimately results in disruption of imprinting across multiple sites across that particular chromosome. So the fact that you're seeing multiple disruptions of imprinting for that specific patients is indicative of patients with uniparental disomy. So in addition to picking up the common imprinting disorders, we can also pick up uniparental disomy fairly easily. And many of these patients with developmental intellectual disability currently get a microarray as a standard tier 1 test. Oftentimes these disorders are picked up by copy number microarrays. Sometimes they're not. UPD, for example, in some cases it's not. So these methylation arrays follow a very similar principle, is that you run these arrays in that particular population and you rule out a bunch of different syndromes that sometimes have phenotypic overlaps and so on. That was the ideology behind kind of doing the initial validations on this. And what we started seeing is in this initial cohort, about 400 or so individuals, that were picking up patients with imprinting defects in loci other than the known loci known to be associated with clinical conditions. So there's a couple of patients with an imprinting defect, this particular locus, and another couple there. At this time, we don't necessarily know about evidence of clinical associations for any of a few hundred bona fide imprinted loci across our genome. Part of the reason why is because the vast majority of them probably are not associated with imprinting disorders, but also it's because we haven't studied them routinely. I tend to think about this in the context of microarrays. So I trained at Baylor College of Medicine, and it was one of the labs that launched the initial copy number microarrays about 10, 15 years ago now. And when they initially launched these microarrays, they launched them to replace a bunch of targeted testing that they used to do by FISH for the common microdeletion and microduplication syndromes. When they replaced this test with a microarray test, at that time, we couldn't interpret the vast majority of the rest of the genome. And fast forward, years from then, that lab alone, I think, has been involved in publishing more than 200 papers, including, identifying, dozens of new microdeletion duplication syndromes. And how they've done it is by introducing a technology in a clinical setting and matching it to the clinical outcomes or phenotypes in patients, and fully then kind of creating associations that really require that scale of data with clinical outcomes associated with them. I anticipate that introducing whole genome methylation profiling in kind of broader assessment of patients with neurodevelopmental conditions and so on, and matching that in a clinical setting to clinical outcomes may ultimately in the long run yield in resolving some additional disorders and learning new things. Another commonly ordered test in genetics labs is Fragile X. It's essentially ordered in almost all males with intellectual disabilities, one of the most common causes. It's caused by the trinucleotide repeat expansion of the FMR1 gene promoter. What happens is once that trinucleotide repeat reaches 200 repeats, the promoter CpG islands becomes hypermethylated, causing the loss of the expression of FMR1 gene on one X chromosome in males. And essentially, you can use whole genome methylation profile to detect essentially patients with Fragile X males. And what I'm showing you here is a large number of males with Fragile X, hypermethylated relative to complete hypomethylation that we see in normal controls. We can easily pick up mosaics. What we cannot pick up is premutation females, and that still requires targeted molecular assays. So when patients are screened with this type of assay, we then still need to do, in subset of patients where we do detect Fragile X, usually these are males that are probands, perform kind of family studies using targeted molecular assays to look at repeat expansions. So the rest of the talk today is really going to kind of, focus on things outside of the things that we knew. And we knew way back when that imprinted disorders are caused by methylation defects, or you can use methylation defects as biomarkers for these disorders. What we didn't know much is anything much beyond that in a constitutional setting. So this is from a talk I presented back in 2014 at a Great Lakes Chromosome Conference. This is where kind of, in my head, a light bulb went off. So we screened about 400 patients that had developmental intellectual disability, and they were tested with microarrays in the lab that I worked at the time. And we performed whole genome methylation analysis, and we started seeing that, of course, we could pick up some of these patients with these imprinted disorders and so on, but we started seeing these outliers. And when you see outliers for any data set, you immediately think, well, there's got to be some sort of technical error, or there's some sort of an issue with data or whatever. And we started seeing that where you could see DNA methylation profiles that are vastly different in that one individual compared to everybody else in that cohort. And what you're seeing here is a bunch of lines essentially represent methylation levels across CPGs in a particular region, across 300, 400, 500 individuals. But you can see in this case, a couple of individuals that deviate. So one of these individuals was a patient with NSD1 mutation, a patient that had Sotos syndrome. And then second patient was a newborn with dysmorphic facies, failure to thrive, developmental delay. And based on this data, we assume, well, if this is real, the patient should also have Sotos, and we sequenced the NSD1 gene, and it turned out that actually the patient had NSD1 mutation. So as a first instance in our hand where we could use the DNA methylation profile practically to predict what's happening at the genetic level. And this is really where, we started kind of asking these questions of, well, how prevalent is this, right? So a lot of the work that I've spent, kind of focused on in the last, few years was to try to map out syndromes where there is a high suspicion for evidence of this epigenetic change. And what we initially focused on was chromatinopathies or disorders of chromatin remodeling. There was at that time, a few years back, there were a couple of really nice papers that came out, including this one from Björk's group out of Johns Hopkins, where they listed a lot of these genes at a time that were known to be involved in chromatin remodeling. So we systematically went after these genes, and we contacted collaborators across the world, because these are rare disorders, so required large-scale international collaboration to collect these cohorts of patients to see whether or not these patients truly had DNA methylation signatures. And lo and behold, you can see here, and this is some of the papers that we published, some of the kind of key ones as we over the last few years. And what I want to demonstrate here is that a lot of these genes are genes that are involved in chromatin remodeling, DNA methylation or DNA methylation mechanisms, and so on. We've expanded, since then, we've expanded beyond those conditions, but now I'm going to really focus on two or three publications where I really want to kind of highlight a few key principles as to what this is. So in this paper we published a couple of years ago, we asked whether or not we can use machine learning on epigenomic data to develop sensitive diagnostic episignatures. We had 14 different disorders that we studied with cohorts of patients, and we had hundreds of normal controls for which we had whole genome methylation profiling done using these methylation arrays. We developed a support vector machine-based machine learning protocol to identify these episignatures, and then ultimately wanted to test this protocol for identification of patients with the underpinning genetic conditions and VUS assessment. I'll talk a little bit more about it. So this is a circles plot from that study. About 8 or 9 of these conditions were found to truly have distinct DNA methylation profiles. Circles plot here on the left side shows you an overlap in the probes that are identified as differentially methylated. So as you can see, for many of these conditions, so Sotos, DNA methyltransferase 1 neuropathy, ATRX, most of the probes are unique to that particular condition, but there's certainly an overlap. At the time, we couldn't necessarily make a lot of inference as to what that overlap meant, functionality, et cetera. But the key was that there were enough probes that were differentially methylated. We can start really thinking about using them as biomarkers. This is a correlation plot showing instances where at least 3 probes are evident, and showing that in some cases there's an inverse as opposed to direct relationship between the DNA methylation profiles. And what I mean by that is, if you look at any one of these regions where we see these differential methylation profiles in these conditions, we can see a relevant relative either hyper or hypomethylation between controls in these disorders. So an example of this particular region in the promoter of PRDM9 gene, where 2 different disorders have significant changes in DNA methylation. So ATRX syndrome in blue and Sotos syndrome in green, and reference normal controls are in purple. And what I'm showing essentially is a relative hypomethylation in Sotos patients, and then further hypomethylation in patients with ATRX. So using, I'd say, fairly robust quality control followed by methylation detection analytical protocol, and on the right-hand side, we've listed some of the steps that are involved, but you can certainly pull out the paper and get a bit more details on that. We developed what we refer to as a methylation variant pathogenicity score. And essentially, this is a set of probes and a signature that allows us, based on DNA methylation profile, to identify patients with that specific condition. So instead of, say, sequencing a gene based on a DNA methylation profile using this score, we can tell whether or not a patient has ATRX, for example. Because when we apply the ATRX score, only patients with ATRX score very high, whereas patients with all the other conditions that are listed here, Kabuki, Sotos charge, etc., don't score very high. And you can see that for each one of these conditions, it's highly sensitive and highly specific. And this, at this point, was tested against a cohort of about 3,000 individuals that were that they were controls, and it was essentially demonstrating 100% specificity in those cohorts. So before we get into what can we do with this, I will have to mention that at this time in this study, in white were the disorders that we could identify an epi signature, and in the yellow and the red are the ones we couldn't. I'm highlighting Coffin-Cyrus syndrome here because at that time when we published this was early in 2018, we were not able to identify a unique epi signature. Now, fast forward about 6 months later, we published a follow-up in that same cohort where we said, no, actually there is an epi signature. And what changed was that we increased the resolution of these methylation arrays. So previous study was done with primarily 450K arrays. And this one was done with 850K arrays, so more probes, more sensitivity, and we increased the number of patients. But the way that we increased the number of patients that went in the cohort was because, again, these are rare genetic conditions. It's really hard to obtain, specimens that are positive for these conditions. We recognized that ARID1B, which is what was tested in the previous study, was part of a complex referred to as a SWE/SNF chromatin remodeling complex. And patients with these, what's referred to as buffopathies or BAF complex buffopathies, have fairly similar clinical features. So they have Coffin-Cyrus syndrome type 1, 2, 3, and others, and then also Nicolaides-Baraitser syndrome. So we combined them all together, and lo and behold, we found what was at the time, the first protein complex related episignature. So essentially all of the patients with the mutation in any one of these buffopathy genes cluster on the one side relative to the reference controls. We could tell there were some subtle differences between these signatures. By the time, we simply did not have high enough numbers of patients to be able to lineate these. Since then, we've tested probably 30 or 40 additional patients that were positive, and we're starting to further refine. So we're now actually getting better at pinpointing exactly which gene within that complex. So when we see an epi signature, we can tell beyond whether or not it's a buffopathy, rather if it's a Coffin-Cyrus type 1 or type 3 or Nicolaides-Baraitser, which helps us then in focusing on which specific genes we need to be doing the testing to confirm this. But one of the biggest, I think, clinical impact pieces of data from this study was from a cohort of patients that were submitted to us for the study, through collaborators. They had previous genetic testing where the genetic testing was inconclusive. So the banes of our existence, the VUS, is variants of unknown clinical significance. So using the epi signature, so this was the genetic testing for these various mutations or variants, and previous clinical reports that we issued as pathogenic or likely pathogenic or VUS, we were able to very clearly segregate them based on the DNA methylation, either pathogenic or not pathogenic, systematically across this whole cohort. So this is where, for the first time, we're really starting to kind of think about, can we really start using this in a clinical setting, more broadly, and next couple of papers, I'm going to kind of focus on that. But the other thing that I guess is important to mention is that, we have patients that have VUSs in genes for which it's very difficult to establish a diagnosis. Classically and historically, it would require family segregation studies when they're available, demonstrating that it happens in more than one family, more than one independent family, or doing kind of very, complex, sometimes, and sometimes it wasn't possible, complex functional studies to demonstrate kind of functionality of these variants. So now we're in a position where we have essentially what is a functional assay in patients' blood, in patients' DNA per se. The same DNA that we do the genetic testing has in it DNA methylation profiles, which are a functional consequence of the underpinning defect associated with that particular gene. So it becomes an immediate functional assay, and a most direct functional assay you can really imagine, because it's in that same patient and on that same DNA specimen. What's really interesting also from this paper here, and without going into a lot of details, so testing versus validation cohorts, all of the positives, negatives behaved exactly as predicted. So we had essentially 100% sensitivity and specificity across thousands of individuals for being able to detect. But there was a discovery cohort here where we screened a whole bunch of patients, a few hundred patients, that we had in the database, and applied this bafopathy signature algorithm on them, and 2 of them came up high as having bafopathy. So we went back and sequenced them. One of them actually had exome sequencing and was negative, including ARID1B. But we actually went and did a bit of deeper dive and did targeted panel assessment, turned out actually the patient actually did have an ARID1B mosaic complex mutation that was missed by exomes. So this was the first instance where we could actually really, demonstrate very well that we could actually use these epi signatures, even in patients that had exhaustive genomic testing, including exomes, to ultimately pinpoint the specific genetic underpinning cause. The second patient, to this day, we still don't have a specific mutation identified. So this now begs a question, and the patient clearly has physiological features associated with the Coffin-Starr syndrome, or bafopathy complex. If we're not detecting a genetic mutation in the known genes, can we have something as an epigenetic diagnosis, right? And currently, we don't really have guidelines or regulatory around this to even be able to think about it, but certainly it's where we want to go next with this technology. Because we're currently not sequencing introns, we're not sequencing 5' and 3' UTRs of genes. Even if we are, we're not able to interpret them very, very, very well. This is one area where this type of testing can certainly help. So in many of the papers we published over the last little while, we used to always get questions from the reviewers about what happens if you apply this algorithms. And at the time we had about 14 signatures identified. Today we're close to 55, but at that time we applied these signatures to a cohort of about 965, almost 1,000 patients that had exhaustive genomic testing, exome sequencing, arrays, targeted panels, with presumed hereditary genetic conditions where they did not have a conclusive answer. And at the time, as I mentioned, there's I think about 14 or 15 different syndromes that we could actually very uniquely identify based on these epi signatures. So this is a 2-dimensional PCA type plot that demonstrates really nice segregation of these signatures away from each other in both testing and validation cohorts. We are at this time now in the database of almost 6,000 individuals that we've tested at a time. We had up close to about 4,000. And what I'm showing here again is this methylation variant pathogenicity scores for each one of these conditions, demonstrating high level of sensitivity and essentially absolute specificity across 3,000 or so reference controls. In the same study, we had a cohort of patients with VUSs or very strong clinical presentations associated with specific conditions, but they did not have a genetic diagnosis. So essentially, and I chose this plot to just highlight a couple of interesting features. It's a cohort of charged patients when we apply these MVP scores, you can see the subset of them scored positive for charge, so they're now diagnosed. The VUS was reclassified to having charge. And then the subset of them that scored as none of these signatures, so they did not have charge syndrome. However, one of the patients turned out to have a epi signature that is not charged. It actually was an epi signature associated in ADMP1. We went back and sequenced this patient. Actually, a physician that submitted a patient sequence, and turned out that they found mutation ADMP gene. Why is this important? Well, because as we expand a number of these conditions, we do the testing for, many of these conditions have very similar kind of clinical presentation and phenotypes, and physicians oftentimes will order testing that matches what they think to be the best fit for their patients, but that's not always the case. And when we do screening like this, we can pinpoint to the underpinning genes, much more specifically, so the confirmation to be done. And we have dozens and dozens of instances where we've done that effectively now. We've launched this testing, and I'll talk a little bit about this clinically in Europe and US just within the last year, and we have more cases that have been resolved like this that I can kind of put in any one of these presentations. So another example, Class-Janssen syndrome, a number of patients positive Class-Janssen's, one that was negative, one that had Soto syndrome was confirmed. Similar case for a cohort of patients with Kabuki, one with Class-Janssen syndrome instead. There were 13 patients in that cohort that had distinct epi signatures that were associated with one of the conditions at that point, which we had mapped. For the ones that we could obtain DNA and REBs to do sequencing, we've gone on done sequencing, and a number of them actually picked up specific genetic mutations that match those epi signatures. They were either not detected or missed or just were not tested. In this cohort, we identified a significant number of patients that had other conditions, including imprinting conditions like Prader-Willi arrangement, Beckwith-Wiedemann, and there's a patient with a couple of patients with Fragile X, and a few other patients, some of the other imprinting conditions that were they're now recognized to be pathogenic. But you can look at the paper in more detail to find out, a bit more detail about that. But what's also interesting in this paper, and this is some of the work that Andy Sharp in New York has been, publishing a bit on now and presenting on more systematically. But we're starting to kind of, as we're now developing these larger databases, look at the phenomenon of epivariance. So epigenetic variation that's regional, that's specific to a particular underlying SNP. And in this cohort, we looked at epivariants, so unique events. So I'm showing you an example of one. So unlike these episignature, we see genome-wide disruptions that cumulatively form an episignature. We're also seeing these single events that are at single genes and so on. It's an example of a patient with promoter hypermethylation, so essentially hemimethylation. One of the two alleles is methylated, whereas 3,000 or 4,000 individuals in references here are not methylated ever. And it happens to be a promoter of the VRK2 gene. This is a young boy with idiopathic generalized epilepsy seizures. At that time, he had previous testing, he had a 3' UTR, 30 kb upstream actually microdeletion in the genome close to that gene. At that time, it was reported as a VUS. Since then, there's been publications that demonstrate VRK2 is through GWAS as an epilepsy locus. So this particular region also happened to have a hypermethylation of the gene promoter on that allele. So that is possibly indicative loss of the expression of that gene. Now, at this point, we don't have enough data to be systematically able to say, yes, this is the cause in this patient. But as our databases go from, thousands to tens of thousands, we might have enough, statistical strength to be able to make these type of associations, particularly when we associate this clinical condition. There are a number of other patients where clinical phenotype matched these very rare epivariants in specific genes that if we were to identify a mutation in that gene, they would be diagnosed with that condition. And you can read the paper to kind of get a sense of, how many of these we've seen and what they were. And then the last paper I'm going to focus on is the most recently published one earlier in this year, where we really broadly expanded. So we're now systematically mapping these conditions. We have a network of collaborators across the world, really, that are, collecting these patients and patient cohorts. And what our goal is really is to map out as many of these conditions as possible, because of course the larger number of genes that we identify epi-signatures, the broader utility of something like an epigenetic test, and the better case we have for introducing this more broadly for screening of patients with developmental intellectual disabilities. And I'm just showing as a comparison, just 2 years separated Circo's plot from the 2018 paper versus the one in 2020. So you see many more conditions being added, some overlap. But now that we have a larger number of conditions, we can start looking at the overlap probes and start making some sense as to whether or not there is some interesting functionality in these overlaps. So as an example, one of the plots we've done here is a clustering analysis, essentially a dendrogram, a relationship dendrogram based on the similarity of the DNA methylation signatures across these conditions. And without going into a lot of detail, I'm just going to focus on the branch here on the right side. All of the conditions on the right side branch have hypomethylation as a key feature in their signature. And they're all disorders where the primary clinical feature is disruption of growth. The one branch is overgrowth syndromes. The second branch is growth retardation syndromes. So it's really interesting and I do actually believe as these data sets increase, the epi-signatures and correlation of these epi-signatures, they're underpinning genes and gene regions, might start telling us some things about functionality or what those epi-signatures actually do. They're very robust biomarkers, which is the primary goal of a lot of the research that we do. But I do think there's a bit of biology that remains to be uncovered, as we map more and more of these things. And again, just showing an updated PCA plots demonstrating essentially the nice separation between these conditions. And in some conditions, actually, really interestingly, and I again don't have a lot of time, we have a split in epi-signatures. So we have conditions where we have more than one epi-signature, depending on which domain the mutations are happening. In some cases, they overlap specific clinical phenotypes. In other cases, they're not, or not as much as we can define it right now. We are seeing clustering of similar conditions, like as I demonstrated, kind of the buffopathy complex having a very similar DNA methylation profile, but we're starting to see some subsegregation between the different genes. So there's a lot more to be uncovered. There's 3 or 4 studies that they're in the process of being published where we use these epi-signatures in novel disorders as functional evidence that they're new disorders. So cohorts of patients with mutations in a novel gene where physicians are trying to make a claim that this is a new condition. We've run the methylation profiling and demonstrated a unique epi-signature, which really kind of bonified that this is likely an independent entity that's not just some alternate type of condition. So what do we think is happening here, just in general sense? Well, when you have normal development, usually you have normal gene, constituency, and ultimately you end up with normal phenotype that is reflected by the normal, epi-phenotype. I used to call this epi-genotype model, but really I switched to calling it an epi-phenotype model because it's not the cause, it really is a consequence of the underpinning genetic changes. If you have a mutation in one of these genes that has a consequence of an epi-signature defect, you will ultimately end up with an abnormal development, and depending on what gene it is, you'll end up with a DNA methylation profile in peripheral blood. Other tissues as well, we have some evidence now that is unique to that condition, and again, these epi-signatures are going to be somewhat specific depending on specific gene that is involved. We just submitted another paper and review. This is a kind of an up-to-date picture. We got about 55 genes that we've mapped so far. Many of these genes have phenotypic consequences of disorders where there's a significant amount of phenotypic overlap, making diagnosis of these disorders very, very difficult. Of course, there are hundreds, if not thousands, of VUSs that are being uncovered in many of these genes across the labs, across the world, regularly. So we're certainly hopeful to be able to kind of ultimately resolve a lot of these as a result of these epi-signatures. I will say that we have launched this testing clinically in partnership with, at this point, 2 labs, one in the US and one in Europe. We're certainly looking to expand this partnership more broadly. The initial launch of the test was to really use it in clinical setting for resolution of patients with variants of unknown clinical significance. Now we're starting to see it be used a bit more broadly for screening of patients with, their unresolved bioexomes and others. This has been now going for about 7-8 months. We've just recently updated our test to EpiSum version 2, and this is all the new disorders highlighted in a dark highlight here that have been added since the EpiSum version 1. The way that this partnership works is the testing is done and reporting is done by individual clinical labs in US and Europe. Greenwood currently for US market, and then AUMC for European market. The data generated on site comes to us through file transfer protocol into our clinical portal, and the data is processed, analyzed, and reported back to those labs that are then issuing reports back to physicians and patients. We just announced a national study. We just got funded through Genome Canada to look at implementing this technology as part of the routine screening for patients with developmental intellectual disabilities with strong suspicion of genetic component. These patients currently get microarray testing primarily, so there's be a lot of these patients will be patients that will get this test as a primary screen. Subset of these patients will get it as a secondary screen after failed or inconclusive genetic testing, and then another subset of these patients, about 2,000 actually, are going to be reserved for identification of epi signatures in up to about 100 additional syndromes. So we're aiming to get about 20 patients per condition as part of the study. And this is just kind of a breakdown. So we have a discovery cohort, about 2,000 patients. We're looking at about 100 additional signatures. We're aiming for about 20 patients per cohort. And then for looking at the diagnostic utility impact, first year about 1,000 and another 1,000 as a reflex or unresolved. The study is supposed to run for about 3 years. It's through a part of a genomic application partnership program. Genome Canada funds these studies that they consider to be close to clinical implementation. So there's a big kind of health economics aspect embedded into this study as well. And we're participating or essentially partnering with Care for Rare for the recruitment of all these patients, because it's the same patient demographic that we'll be looking at testing. And we're supposed to already launch this study, but due to COVID, it's been delayed for a couple of months. And finally, I'll acknowledge many leads of the many collaborators across the world. Certainly, I couldn't list everyone on there, but this type of work really takes a village. Rare genomic disorders are rare by definition, so to pull together resources that identify patients and patient cohorts requires really a global partnership and global collaboration. And certainly, again, of course, I have to thank the patients that are involved in these studies. And finally, thank you for your attention. And hello from the time pre-COVID. This was the Christmas lunch in our lab. Thank you very much, and please take any questions.
Thank you. You don't get to hear the applause, but everybody can virtually applaud. So, that was really fascinating. I think, Piotr, you'll be able to see the chat to see if there's questions. But maybe while people are...
Yeah, if they raise their hand here virtually, we can unmute them and kind of try to make it more interactive this way.
Great. Maybe I'll start with the comment/question as people are raising their hands and compiling. I think what's, in addition to the science that's got into this, what's so impressive, the ability that your group has to go back iteratively and sort of go back and see the phenotype. So you see an undiagnosed case, you find an interesting pattern, and then you clearly have to go back and reconnect with the patient or the physician of the patient. And so I wondered, we've been trying to set something up like that here at Boston Children's with our genomic cohorts initiatives, but I wonder if that's something that's unique to your institution or if that's a Canadian-wide thing. It's really quite a paradigm for being able to really do meaningful new discovery.
Am I unmuted? Okay, I think I'm unmuted now. Yeah. So so many of these patients that we've reconnected with now are reconnected through our clinical programs. So essentially, when patient gets tested through either one of the clinical sites that is currently doing the testing for this, part of the protocol is when you identify these epi signatures, of course, is to report a result back to the physician, and physician then will decide as to whether or not additional testing will be done. In some cases, for example, Greenwood will perform their own genetic testing to confirm as part of the test itself. Depending on the study, and as I mentioned, there are many different sub-studies that are part of these larger cohort studies. It is really dependent on a particular site and particular REB that was involved. In some cases, we are able to reconnect, and in other cases, we're not. Most of the studies that I described here, our current REBs are designed so we can actually go back and issue the result back to the physician, who's then able to connect with the patient and provide those results back.
Good.
And it's really just an REB related question. We designed our studies so we can, based on these findings, report results back to physicians, who then at their discretion are able to perform genetic testing to confirm these.
Great. Thank you. I think we've got a bit of reverberation, but Piotr, shall I mute myself and you can see if you've got other questions?
Yeah, so David Miller, do you want to ask? I just, I don't know.
Oh, okay. Yeah, my question was whether you foresee a model. I could envision a model where if you characterize enough conditions with this signature, it might be more cost effective to start with this as opposed to exome sequencing or a microarray or something like that.
The model we're testing in this national trial, and I absolutely agree with you. I mean, you know, the health economic aspects of this are, you know, not unnoticed. The array technology that we're currently using for this is, you know, comparatively inexpensive. These arrays are about $300 per specimen, and certainly much less of a, you know, cost than performing, say, whole genome sequencing, whole genome trios. At this point, based on the number of conditions that we are able to detect, we're estimating about 3 to 5% diagnostic yield in that patient population. Now, you know, as the, you know, number of conditions grows, we expect we may be able to detect more, and it's really going to come down to as to whether or not the additional prior diagnostic yield prior to broader genomic testing is going to be cost effective. I expect it will. And the other thing to consider also is that we are also able to detect patients, of course, that even post-genomic testing, we don't have answers for. About half of the patients, even with full genome sequencing now, don't have a resolution. And oftentimes it's because it's these missense variants that we can't interpret, or it's variants in gene promoters and introns and so on that we're not able to interpret. So doing this as a priori allows you to kind of pinpoint a specific region of a gene or a genome to do further assessment in, and to do to kind of focus the genomic testing in. And the other kind of point I want to make is currently a tier 1 ACMG test for patients with neurodevelopmental presentations is a microarray test, developmental delay, and so on. So that's one of the first tests that's done in these patients before they go on to do broader testing. So we've been in discussions with Illumina specifically. There are potential, so our lab, for example, runs the Illumina copy number arrays for all these patients first as a standard ACMG-based protocol. So these copy number arrays are about 700 or 800,000 probes, and methylation arrays are about 800,000 probes. So, you know, technologies are capable of printing, you know, a few million probes on each one of these chips. So it might be possible to combine the copy number and methylation capacity on a single chip. So instead of just ruling out copy number alterations, ruling out copy number alterations and methylation disorders as part of the initial single test. Of course, the challenge here then is that we're having copy number alterations that are able to be detected, and essentially everyone has capabilities now to interpret these findings. The DNA methylation component is something that we don't necessarily have, you know, as much of those capabilities until the databases become larger, until we map out as many conditions as possible and find a way to integrate this data into these platforms and share this data, you know, more broadly. But I do think it's got potential to go in that direction.
Good. So I have more questions. So from Jeff Wall, I'm just going to unmute you right now. Jeff, you can ask your question.
So really nice talk. I wonder, how much does methylation change in healthy individuals through the age spectrum? Is this a situation where if someone had methylation as a young child versus a young adult, would we expect to see changes? And do we see methylation differences between healthy individuals based on ethnic differences?
That's a very interesting, very complex question. And in short, absolutely yes. We do see changes in DNA methylation in peripheral blood and other tissues that are age-related. As part of our analytical algorithms, we essentially control for those changes. There are many other changes that are in peripheral blood. They're related to other things like, for example, sex, ethnicity, as well as the blood cell composition. So there are currently ways to deal with and normalize for that data, prior to the assessment. When we identify probes that are, part of episignatures, part of the process is to identify probes that are least susceptible to that change. I can say that we published a couple of papers. We actually demonstrated the evidence of accelerated aging clock and DNA methylation in some of these conditions as well. So it certainly is something that is fairly complex. It's not static across the genome. There are certain sites that are more susceptible to these changes, and we can identify them and we can control for that. And when we identify these episignatures to look for regions that are least variable. Between individuals, if you look at DNA methylation profiles, even with these methylation rates, looking at about 850,000 sites, we see quite a bit of inter-individual variation. That's maybe accounted by some genomic composition, but also maybe accounted by other changes that are inter-individual variabilities. And what's really interesting is that a lot of this happens in gene bodies. So there's quite a bit of variability in gene bodies. However, CPG islands and CPG island promoters in particular tend to be fairly consistent across individuals. So all these, regulatory elements that have known kind of function in gene regulation and so on, variability is minimal. In the rest of the genome, there's quite a bit of large variability that's really not accounted for. And those are some of the complexities we deal with when we create these kind of machine learning based signatures, is to try to pinpoint probes that are least susceptible to these type of effects, but we see them.
Okay, good. So thank you so much. And I have another couple of questions from Philip. I'm just going down to unmute you. Yeah, you should be unmuted now.
Yeah, hey, this is Philip Boone. Can you hear me?
Yes, we can. Yeah, go ahead.
Great. I like this talk, and I enjoyed reading your papers as well, Bekim. My questions are, when I send samples to Greenwood for the EpiSign array, for example, do those get reanalyzed and reported out over time if a new episignature is identified? In the meantime, would you personally submit, kind of with microarrays, I think as a clinician, it's very valuable, of course, that VUSs get reported out. Would you report that VUSs from this technology be reported out? Or I think it's more appropriate to do what it seems they've been doing, which is only reporting out if there's a specific confirmed known episignature that's identified.
First question, because we've gone now on to the EpiSign version 2, as part of the protocol, both in Europe and US, we've established that we are currently rescreening all patients essentially with these algorithms as they get added to the new episignature. So there's a clinical part of this test, and there's the kind of R&D discovery part of the test. So certainly, while a lot of this work is in publication discovery, we're not applying it to clinical samples. Once we upgrade the test and we are performing inter-site clinical validations as per CAP, CLIA, and so on, then we apply the new signature onto the previous samples and then identifying the patients. We've seen a few already where they weren't detected by the initial version of the test, and these are then at the discretion of each lab reported or not reported back to physicians. We're trying to create some sort of a generalized agreement as to how we're going to deal with this. And I do think that, and part of this Genome Canada trial, we are looking to establish kind of an international consortium of experts to start really talking possibly to ACMG about drafting some guidelines around use of this technology in a clinical setting. As you know, this is very new, and we'd like to have some sort of a systematic guideline, internationally for how this is to be used in the future and so on. And then the data. So this is interesting. So we are seeing, vast majority of the cases that we test currently, we're testing to either resolve patients with variants of unknown clinical significance. Sometimes we're testing to screen patients that haven't had genetic testing, but there's strong suspicion. And we're in about 95% cases able to give a high confidence report. It's either that the patient matches a particular epi signature or not. I would say about 2 to 5% of patients we're seeing right now is maybe closer to 5%. We're seeing somewhat of low confidence results and low confidence scores. That can happen for one of a number of reasons. We're now seeing that for some conditions, the DNA methylation signatures are not as robust in all individuals. And we're not necessarily certain as to whether or not it's because of age effect or because of variable phenotype or penetrance of a specific mutation and so on. So we're learning as we go along. So a subset of these patients, we give reports where it's a low confidence report for, say, a negative result. We're also issuing a low confidence report for a positive result. And in some cases, it may actually be due to mosaicism. And we're starting to kind of accumulate this data, get a good sense on it. And then we are seeing now more and more genes where we have subsignatures related to either specific type of mutations or specific mutational domains. And it's really fascinating. It's kind of unraveling as we collect more of this data. There's off the top of my head, probably about 5 or 10 different studies. We're currently involved with various collaborators where we're mapping out distinct domains. And what happens is if it mapped an epi signature based on what's the most common type of mutation that's in the literature and where we can collect the patients, and we omitted, say, a particular domain where the mutations are very rare and they cause a separate epi signature, possibly a separate syndrome, it may mean that there will be a false negative. So if you get a negative result for an epi signature for a particular condition, while it should work for vast majority of the patients that suffer with this condition, we're unraveling more of these kind of subtypes. They're more rare. They may actually not have the exact same epi signature. Sometimes the epi signatures overlap, so what you end up with is a kind of a low confidence score. So it gets a bit messy for I'd say about 5% of patients we're currently testing. I think it's going to get messier as we add more conditions, but as we add more patients, it's going to be interesting.
Thank you.
Well, thank you. I know some people probably have to go at 1, but if you have a few more minutes, I know a few people have stuck around. Piotr, are there other questions that have come in?
So nothing from the comments here. I know you had a couple of questions.
I do. Thank everyone else for coming again. I don't want to hold everybody past 1 if you have virtual patients or so on. But Bekim, if you have a minute, I just wanted to ask you one other question and follow up. Although I have a few, but I was wondering about going back to your initial comment and your note about the limitations of different methylation patterns across different tissue subtypes. And it's very much reminiscent of any sort of somatic mutation study that we've done, whether it's the brain or other organ systems. How similar are the methylation patterns between blood and brain, for example? Because it seems like the methods that you're using, even looking at blood, are detecting with pretty good fidelity some of these very specifically neurological disorders. Piotr, can you unmute Bekim?
Can you hear me? Yeah.
Yeah.
Okay, good. So I'll give a couple of different angles to the answer to that question. So we've done very limited amount of work in other tissue types, and we've done it in the context of fibroblasts and buccal swabs. We've demonstrated, or we've seen essentially, that there is a overlap in these signatures. They're not identical because the alternate tissues oftentimes have differences in baseline DNA methylation profiles. So the algorithms that are trained against a certain reference in peripheral blood. So what you end up getting as a result of these epi signature scores is not going to be perfect, right? But we do see almost without exceptions where we looked at, they're somewhat evident across all tissues. So we think it's a result of an early event that happens in chromatin modeling and the functionality of these genes that kind of echoes across tissue types. Whether or not it has a function in peripheral blood, because most of these patients don't have a hematological phenotype. That's for deliberation, but it certainly is being able to be used for surrogate. But I do think that there will be conditions where these effects may actually be targeted to specific soft tissue type. I think of one that really bothers me to this day. We are a regional reference lab for RET testing, MECP2. It's a methylbinding domain gene and so on. So this is the one we thought for sure is going to be that signature, except this is a reader as opposed to writer or remodeler type protein. So we've tested probably the largest cohort of all patients we've tested so far with RET, and we couldn't find anything, not in peripheral blood. And clearly patients have very distinct phenotype. In this setting, it would be worth doing or trying to do testing in kind of more direct tissues like brain, but as you know, it's really hard to obtain these type of tissues and get a sense. And on the other hand, even if we demonstrate as an episignature, just from the practicality standpoint, what we're trying to do is develop biomarkers that we use in a clinical routine diagnostic testing. It becomes really difficult to implement for routine testing as well. So it's kind of a double-edged sword, really.
Well, that's great. Thank you again. This was really fascinating. And hopefully we'll be able to stay in touch virtually, if not see each other soon in person.
Well, thank you very much. We have a session on this at ASAG. There's a number of people in an invited session that's really focused on this work. 4 or 5 different researchers going to be presented on this. Hopefully ASAG still goes live, and if you happen to be there, happy to meet and see any of you there.
Great. Well, thank you again, and stay well and stay warm in your snowstorm.
Thank you. It stopped.
That's good.
Thank you very much.
Well, take care. Have a
Bye-bye.
good afternoon. Thanks, everybody.